Good afternoon, everyone. We are from the European School of Frankfurt, and we'll be talking to you today, Vincent Archer, Diego Soto, and myself, Hamza Salim, about an investigation to determine the short-term effects of flavonoids on adolescent prefrontal lobe activity.
So you're probably wondering what flavonoids are. Well, flavonoids are a group of organic compounds that are commonly found in plants. In these plants, they serve several purposes, such as the colorization of flowers and fruits to attract seed dispersers and pollinators, and they also filter some UV radiation. So there are multiple kinds of flavonoids. The ones that we focused on are a subgroup of flavonoids called anthocyanins, pictured in the middle, and these anthocyanins are themselves classified as isoflavones, top right. Now there are several others flavonoids, and their basic molecular structures are pictured here. Now, anthocyanins can be found in various fruits and vegetables. We focused on blueberries, however, because they are very rich in anthocyanins and they don't really contain very much of any other kinds of flavonoids. Pictured here are some blueberries and, again, the basic molecular structure of flavonoids. Now, the parameters of this experiment begs the question of why we did it on teenagers. Well, we have to look at how teenage brains work. During adolescence, teenagers go from using their amygdala to make everyday decisions to using their prefrontal cortex. Now, the amygdala can make decisions, but it does so using emotions and impulses. Once you're an adult, however, you make most of your decisions using the prefrontal cortex, which is more logical and reasonable. The prefrontal cortex is a region of the frontal lobe which is located at the front of the brain, and the prefrontal cortex is responsible for logical thinking, short-term memory, and one's ability to concentrate, and some of these traits were tested.
Well, now for the method, there were a total of 28 subjects divided into groups of 14 each, Group A and Group B. And Group A were given the control in the first week and the blueberry juice during the second week. And the other way around, Group B were given the blueberry juice the first week and the control the second week. Well, here you can see a schedule where it shows Group A taking the control for 5 days and taking a test the 5th day, which, well, each participant had a number ID to ensure confidentiality. This is a typical crossover design with a washout. So the first test was a memory test. As you can see here, there are 26 objects. We projected this image for 30 seconds. After the 30 seconds had passed, they had 4 minutes to write down as many objects as they could remember. The aim of that test was to examine the functioning of the hippocampus. It is thought that flavonoids are apt to trigger the protein extracellular signal-regulated kinases A1 and 2 and B signal pathways, and this leads to an increase of neurotrophins. Neurotrophins help the survival and development of neurons in this region, which ultimately improves memory ability. Now, the second part of the trial was a speed test. The subjects were given 4 minutes to complete a series of 22 questions. Half of the questions were true-false type of questions, and the second half were a set of diverse questions. The aim of that test was to challenge the prefrontal cortex, since it has to cope with functioning under time pressure and deciding and working out the answer, concentrating on each question and deciding whether to answer the question or skip it. The last part was a logic test. The subjects had as many time as required to complete a series of 5 questions based on logic thinking. The aim of that test was to check the left side of the frontal lobe, since it has been observed that the solving of logical problem is asymmetrical, meaning that strictly the left side of the lobe is involved in logical thinking. Now, the materials used were the blueberry juice pictured on my right, so your left, which was 30% water, 10% blackcurrant, 20% raspberries, 20% blackberries, and 20% blueberries, and the control on my left, which was 96% water and then 4% of yellow food coloring.
Now, looking at the results, we compared the results, the participants' results for when they were on the flavonoids to when they were on the control. And we see that, on average, the people on the flavonoids did 0.26 better than the people on the control. Then this was their grade of the test. And this is an increase of 2.6%, which is quite impressive for a drug, for flavonoids given over such a short period of time. We also see, it has to be noted that these are average results, and by doing a crossover method, we were able to see exactly how many people showed improvement. And the majority did, with 68% people showing improvement when they were on the flavonoids. Now, for the second test, or the speed test, we see that the flavonoids show improvement compared to the people on the control by 7.7%, which is a quite staggering amount. But we would expect this because, as Vincent said, in this time of adolescence, the brain is undergoing great change, and this region is being developed, so we would expect to see the greatest amount of improvement in this region. Now, it also is true that the amount of people affected should be greater than the one in the memory, and this is also the case, with almost 3/4 of the population showing improvement on the flavonoids. Now, for the logic test, to our knowledge, there's never been any sort of test done with flavonoids and logic, so it was very interesting when we found out that there is actually barely any change on the logical reasoning when the participants are on flavonoids. In fact, you can see that the control has done better than the flavonoids. I know it looks like quite a lot, but that's up to 2 decimal places, so it's actually only 0.5% difference. So we decided to regard this as almost non-existent, no difference at all. And we also see that the amount of people affected is not as consistent as in the other 2 regions that we tested, with only 46% being influenced by the flavonoids. Now, looking at this graph, we see in blue how, on average, the people did on the control, and in green, on average, how much better they would do if they were on the flavonoids in each field that we tested. On the logic, you see they would go down by a bit. Now, here you see the percentage of people affected by the flavonoids, and the most being in the memory and speed, and then the 46% being in the logic. So to conclude, we see an improvement in the hippocampus region, which is the region of memory, and in the prefrontal cortex, which is the region that undergoes change. This would be, on average, which would be the case for 68% of people, and an improvement in their test by 2.3%. So we see an improvement in memory and in speed, but not in logic. So, as Bob would say, it is not logical. Thank you. Thank you.
Judges, anything you'd like to ask?
2 questions. Your sample size, how many students did you test?
We started with a rather large sample size, but we had to disregard many people because if they missed one dosage, we couldn't include include them in the end result. So the final amount was 28 participants.
28, okay. And you talked about one result being only a couple of, down in the second decimal place and therefore not significant, and the other ones you thought were significant. On what basis did you decide whether the change was significant?
Well, we compared them to each other. So we saw that the speed and the memory were around 6-7% improvement, and this logic being just about 0.5 in fact percent, where the control group actually did better. So we didn't think this was anything remarkable. We think it's something to do with it being asymmetrical in the brain, logical thinking, but we would have to put more research into it if it's the case.
Okay, thank you.
Anything else?
I got one.
Could you come?
I'll just say it like that.
Yeah.
So you use juice, right?
You use juice.
Right?
Juice,
and control. How can you tell it's the flavonoids that had the effect and not the sugar?
How can you be sure it's the flavonoids that had the effect, not the sugar?
Well, the majority of blueberries is made out of flavonoids, and there is in fact very little sugar in the actual berry, and in the control there is deionized water and food coloring, and the food coloring is only 4%, so it's very unlikely.
So what is the concentration of flavonoids in this juice? I mean, you said the majority.
What is the concentration of flavonoids in the juice?
We haven't got an exact figure, but we actually took the ingredients used in several other studies.
Yeah, we used the similar portions of flavonoids that they used in Alzheimer's trials with flavonoids and in Parkinson's trials, which we found was an appropriate amount that didn't reach the threshold.
Yes.
I would like to ask, did you tell the test subjects that you gave them flavonoids, or?
No, they didn't know which one was which.
How old were the students?
Well, they were adolescents ranging from, I think, 14 to 17, about.
Did you test girls and boys separately?
No, we didn't do it by gender, we just did it. We just took it as one thing.
Oh, there was one last question over here, then we need to stop. She's saying the control didn't have any taste, so surely people therefore knew whether they had the flavonoids.
And they weren't, we feared this as well, but they didn't seem to notice the difference because it was really quite yellow.
There's just something I didn't understand from one of your slides. The juice that you fed your subjects, it looked as though it was made up of a variety of different fruits.
Yeah.
And I'd understood you were going to test just blueberries because of the one molecule.
No, they all contain this molecule, anthocyanin.
Only that molecule,
Yeah,
anthocyanin.
it's the dark pigment that you see in these berries. We just took blueberries as an example.
Thank you very much. So this brings us to the close of the senior presentation.